uh, just to structure how we're going to go about this talk, I'm going to walk you through how we can look at the foraging ecology of vertebrate species and just looking at how different oiling histories can potentially affect the, the foraging ecology of those species. I will then also focus on how we can look at the energy flow along a salinity gradient that is imposed by different freshwater management um, systems like a river siphon. And then I will then kind of tie that up by talking about the implications for fauna and flora in salt marshes. So if we look at the effects of freshwater diversions and looking at oiling histories and effects of oiling, I'll tie that up and look at how those might have implications for salt marshes. I will then kind of just summarize that and talk to you about how that feeds into the future work that I'll be working with a lot of faculty there at Docs on a recent NAS grant. And hopefully that will then give you a good picture of the types of questions that we're trying to address. So just starting off with the first one, looking at the foraging ecology of co-occurring vertebrates. Generally, if we had to look at this, one of the issues that is always very difficult to grapple with is really the effect of oiling on vertebrates or on fauna as a whole. And particularly, one effect that is always very difficult to assess are the long-term effects. Generally, we know that oiling can have devastating effects. One of the effects can be high mortalities of birds, sea turtles, and marine mammals, like what happened in the Deepwater Horizon. Similarly, we also know that it can alter arthropod abundances and also change community compositions. At the same time, we also know that it can alter gene expressions that are responsible for certain functions like homeostasis and other functions within vertebrates. But generally, if you look in the literature, immediately after an oil spill or a major oil disaster, you have all these studies that are going on, but many of those studies are really looking at acute and short-term effects. Very few of those are really looking at these indirect long-term chronic effects. And considering that, you know, you can have extensive oiling and oil can persist for several years and long periods, it is probably very important to not also just look at acute effects, but also look at indirect and chronic effects over longer periods. But now to do that, as you might imagine, it can be very difficult. So one way that we can kind of work around this is to probably look at using bioindicators as a way to study those acute or indirect chronic effects. Now, why terrestrial vertebrates? Generally, terrestrial vertebrates, if you're looking at the two terrestrial vertebrates that we focused on in this particular work, we focused on seaside sparrow and marsh rice rats. The reason why they're good indicators is that number one, they exhibit very high site fidelity. So what I mean by that is that they remain all year round, and generally they also have very small home ranges. So they do not go very far from where they're resident. And because of that, they're also omnivorous, but at the same time, they also utilize different foraging strategies. So because many of them feed on the ground, therefore they are actually more susceptible to a wide range of environmental perturbations that are happening on the marsh platform. Okay? Now, so what we then decided to do was we decided to ask a few questions. And the questions that we asked, we kind of split them into three broad questions and then worked through those three broad questions to see if we could assess these long-term indirect chronic effects. So the first question that we simply asked was, well, if seaside sparrow and rats are really omnivores, would we expect them to occupy similar trophic positions? And if they do occupy similar trophic positions, what would be the relative importance of, 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 of different resources, for example, aquatic and terrestrial carbon? Thirdly, we also then looked at whether the differences in oiling history, and note, when I'm talking about oiling history, I'm talking about, sites that were historically known to be oiled, because of course we know that oil moves around and oil is redistributed. So oiling history is sites that were known to be oiled. So we then also looked at whether the different foraging strategies, do they really lead to differences in trophic niches. So if for some of you that might not know much about seaside sparrow and marsh rice rats, seaside sparrows are diurnal, and they also kind of they can fly and they can also feed on the ground. So they're aerial hawkers and they're also gleaners. So they can also glean, which pretty much means that they're probably exposed to a larger suite of food that is available to them. And if you look at rats, they pretty much forage on the ground, but they also have the ability to swim. So with that in mind, we were then able to make a few predictions based on something that we knew about their behavior. And the first thing that we knew is that we said, well, they're both omnivores, so it's possible that their trophic positions would be similar. And because rats have the ability to swim, we imagine that they would probably also incorporate more aquatic resources because they can swim. And because sparrows are able to feed on the ground, they're able to aerial hawk, we then also predicted that they would possibly have wider niches because they're able to feed in different habitats. So the approach here was we selected 7 sites that were previously oiled and previously unoiled. And of course, we knew that, of course, there was some redistribution of oil. And of course, that's a huge confounding factor. But here, what we then did is we just looked at those particular sites, and we collected from those sites. So what we simply did is we first collected sparrows. We collected sparrows using mist nets, and we collected rats using baited shaman traps. And because we kind of had an idea that uh, because these two vertebrates are kind of ubiquitous and they're very common, they would be good indicators of what was going on. And because they stay there and they've huge site fidelity, they would at least give us a good picture of what's going on in those sites over a long term. We then collected aquatic and terrestrial baselines, pretty much the basal sources that were supporting those food webs, most likely. We collected rib mussel, we collected leaf hoppers because they feed on C4 plants. We also then collected rib mussels that we know feed on algae. We then also collected the different food sources. We collected POM and we collected Spartina. And the reason why we collected those was to be able to see if those baselines are actually a good reflection of ribbed mussels and terrestrial plants. So if you look at that plot here, what you do see is that POM and C4 plants they definitely separate. And you definitely see that ribbed mussels are definitely closely aligned to POM, and hemipterans are a good indicator of Spartina. So what we then did is we then proceeded to then look at the trophic position. We looked at where they are in terms of trophic level and using rib mussels and leaf hoppers as baselines. And pretty much we used the package called trophic position. And what that pretty much does is it allows you to be able to estimate the trophic level, but also because it's looking at those two baselines, it also allows you to be able to determine the relative importance of aquatic versus terrestrial resources. Okay. So what we then did lastly is we then looked at we then kind of projected all those the isotope values of each of those organisms into into niche space to kind of get an idea of the trophic niche width. And pretty much the idea is that if the wider the niche, so if an organism is feeding on a wide variety of resources, it will have more variation and therefore should exhibit a wider trophic niche. And if an organism is not feeding on a lot of things and is limited in the resources that it's utilizing, it will most likely have a reduced niche. Okay. So what we then did was we then analyzed these results, and I'm going to walk you through some of those results before we summarize them. So just looking at this, where we have kind of combined oiled and unoiled, and the reason why we kind of combined those sites is because we did preliminary analysis to look at different sites between oiled and unoiled. And what we realized was the only biggest differences that were worth noting and were very evident were between the treatment, oil versus unoiled. There were very few site differences within each treatment. So we looked at oiled and unoiled. So this is why they are kind of grouped. So if you just want to look at this, if you look at 2015, so we collected over 3 years. So generally what you notice is if you look at the trophic position, and those stats kind of show you significant differences where I'm comparing sparrows and rats within a treatment. So if you look at this, here what you notice is there were significant differences in trophic position, and generally what you notice is that trophic position generally tended to be higher in seaside sparrow compared to marsh rice rats. And now when you then decide to then proceed to look at the differences in the contribution of the alpha value. So the alpha value here meaning terrestrial contribution. So if we're to look at the relative importance of terrestrial resources, what you'd notice is that in 2050, all sparrows and rats were reliant on terrestrial food. And as you go into 2016, you start to notice differences. So generally, in summary, wherever there were differences between sparrows and rats, generally marsh rice rats were feeding on aquatic prey. And also the trophic position was higher for seaside sparrow. Now, if we then decided to then look at the actual trophic niche that sparrows and rats were occupying and looking at the trophic niche width, this is what we would see. Just looking at the oiled and unoiled, if we looked at at oiled sites, generally you do not have a clear difference. You don't have a clear difference in the trophic niche width. But if you then decide to look at unoiled sites, what you realize is that the trophic niche of rats is less compared to sparrows. Now, we could start speculating, and I'll talk about this when we kind of summarize this, is that the overall trend is that at oiled sites, generally sparrows, rats have an expanded niche, but their niche at unoiled sites is restricted. Now, if we then decided to then just look at oiled versus unoiled sites within species, what you notice is that marsh rice rats at oiled sites are greater than marsh rice rats at unoiled sites. Now, having given you a quick run through of those results, the questions, I'll bring you back to the questions that I asked. And the first question was, do seaside sparrow and rats occupy similar trophic positions? And the answer is seaside sparrows occupy higher trophic positions. And then if we decided to talk about what is the relative importance of aquatic and terrestrial carbon to sparrows and rats, the answer is aquatic food is more important to rats when differences occur. But then remember, in 2015, we had this, it seemed that terrestrial food was important to both of them. And then now the question is, do different foraging strategies in rats and sparrows lead to different trophic niches? Yes, but it depends on whether you're looking at an oiled or an unoiled site. And the other question is then, how do these patterns above differ with oiling history? And the answer is the seaside sparrow niches are greater than marsh rice rats at unoiled sites. And secondly, marsh rice rats at oiled sites are greater than marsh rice rats at unoiled site in terms of niches. Now, having looked at having gotten an understanding that there are some differences in oiling history, and that kind of translates to very subtle changes, especially in the foraging behavior of rats. We can now then talk about something else that we then also looked at. And what we looked at here was we then decided to look at how energy flows along a salinity gradient. Now, we already know that land loss is a huge issue in coastal Louisiana, and it is predicted that we're going to lose a lot of land by 2050. But if you just look at this picture from 1932 and you compare that with 2011, you can definitely see that there's been a lot of land that has been lost. And of course, from a freshwater management, from a management perspective, the idea is to kind of come up with solutions to mitigate land loss. And one of the things that is suggested is that freshwater diversion, so diverting water from the Mississippi and moving sand is a way to kind of mitigate some of those. And generally, if you look at diversions generally will then have already been, it's already been recorded and documented that, you know, they do increase marsh accretion rates, they bring in nutrients, and they increase organic matter. But the one thing that they also do is that they have a huge effect on salinity. They actually affect the salinity. And now we know that changes in salinity, changes in water quality can have an effect on food web structure. And similarly, those changes in marsh accretion, nutrients, and organic matter can also have effect on food web structure. But the question is, one thing that is unclear is what those connections are. So we decided to try and answer this question. Now, talking about food web structure, there are several ways that one can assess food web structure. The one way that you could assess food web structure is you could look at the connections amongst different animals. You could say, okay, this fish probably eats that fish, and that crab probably eats this. Then you could easily work that out. But the biggest challenge with that is when you have to work out all those connections, you pretty much, it can be a lot. One way to look at it is you can, instead of looking at all the connections, you can look at the basal resources that are supporting that food web. So you can look at it from the base of the food web and see the resources that are supporting that food web. So what that effectively does is it then just looks, it doesn't look at all the connections, but then just looks at the two most important sources that we know are important at supporting consumers in salt marshes. So this is one way of assessing food web structure, and that's the way that we decided to look at it. Now, here, the question that we asked was, how does the relative importance of aquatic and terrestrial carbon to consumers change along a salinity gradient that is imposed by a freshwater siphon? And the idea is, if we look at this map, the siphon is up here, and the idea is that because this is where the diversions are happening, you're going to, the water will be very fresh closer to the siphon, and as you move further away from the siphon, you should expect that the salinity should increase. Now, this is supported by measurements that we had done in 2016. You can definitely see that if you look at WPH1, which is really close to the siphon, you can see that it's almost close to zero. And at some sites, it was actually zero, which is completely fresh. So now, having established that there is a type of gradient that exists where you have this increasing salinity, we then had the perfect design to be able to address this question. Now, so what we did is we collected invertebrates, fish, and basal resources. So we collected different invertebrates, from crustaceans to bivalves to insects and all the way to spiders. We also collected fish, we collected different fish, and we also collected the basal resources. Now, as you might very well understand, the biggest challenge when you collect all these organisms in very large amounts, the biggest challenge is how do you condense this into meaningful groups that will not be too tedious to work out. So what we did is we decided to adopt a trophic guild strategy. And one idea is, instead of looking at what an individual organism is feeding on, we can look at what the entire guild. So pretty much just putting them into functional feeding groups. So if you look at these that we have here, we then were able to condense them into functional feeding groups. And the benefit of doing that is, when you put things into trophic guilds, you have the benefit of actually having equal representation at all the sites. So you might not have brown shrimp at one site, but if you use the trophic guild approach, you actually have a very wide representation at all the sites. Okay, so the particular species may not be present, but if you use a guild approach, you have presence at all the sites. So having put those into groups, we also grouped C3, C4, and SAV, which is a shortcut for submerged aquatic vegetation. We also put that into a separate group. And then we kind of split them into carnivorous fish, benthivorous fish, detritivorous fish, and marsh fish. And with insects, we pretty much split them into insects. And then, of course, with gastropods and bivalves, while they're both mollusks, we based on some of the preliminary analysis that we had done, we could see that they were distinct isotopically. So we also then decided to analyze them as separate groups. Now, the analysis, we pretty much also did stable isotope, but we did compound specific stable isotope analysis. And pretty much what this looks at is it predicated on the understanding that essential amino acids are only produced by plants, and animals will need to get those essential amino acids from their diet. And because from plants into an animal's diet, they don't change very much. The change, the fractionation between as they move in trophic in trophic chains, they generally remain unchanged. They're basically very useful as a result. So we looked at essential amino acids, and then we pretty much looked at the different the dietary contributions of the two sources using Bayesian mixing models. Now, looking at that data, if we decided to then just normalize the data and look at it in terms of how things cluster, this is kind of the picture that you would have. Remember, I'm starting from the site with the lowest salinity to the site with the highest salinity. So if you look at this site, and here on your right, this is where you have the functional groupings. If you look at that, what you generally see, the first thing is you do see that there's a clear separation amongst the four sources. So C3 plants, C4 plants, SAVs, algae, they do separate quite well. And then when you then look at, if we decided to look at the fish, they kind of cluster here, probably more of an algal diet. But if you look at things like spiders, they seem to move from being feeding on algae and probably moving more towards C4 plants. Now, if you also look at that, remember the spiders were here, they moved from there, they were here, now they're here. And also there's a small movement for some of the fish and crabs. But generally, what I want you to take away from this is, if you looked at this, generally it seems that as you move from the siphon, there's a general, for some of the consumers, many of them are moving towards a C4 plant-based diet. Okay, now just looking at mixing models, what I did here is I just summarized the aquatic and terrestrial, aquatic being all the algae, the POM and everything, and terrestrial being C4, C3, C3 plants. Now, if we just had to look at it, at just looking at benthivorous fish, if you're looking at that arrow just shows you that I'm looking at increasing salinity. Generally, if you look, there's generally more aquatic contribution when you're looking at sites that are closer to the siphon. But and then if you look at insects, generally, while they do consume more aquatic, generally they kind of maintain this C4 diet. And spiders, on the other hand, you can definitely see that they move from being only reliant on mostly reliant on aquatic food and totally to being more reliant on terrestrial food. Okay, so generally the trend here is that there seems to be, for sites that are closest to the siphon, there seems to be a significant contribution of aquatic food closer to the siphon, and C4 and terrestrial plants become more important as you go further away from the siphon. Now, summarizing that data, our question was, we wanted to assess the relative importance of aquatic and terrestrial carbon to consumers along a salinity gradient. And what we found is that aquatic carbon is more important closer to where diversions occur, with C4 plants making more important contributions further from the siphon. The second thing is we noticed that spiders show the largest change with changing salinities. And partially that's because spiders are at that interface, and generally they're interface specialists. Many of them also have the ability to swim. And then also the question that then, though we didn't really look at this, when you generally at sites that were closer to to the siphon, you did find the presence of more freshwater species. So now the question is, in the long term, what are the consequences of that that might actually cause changes in species composition? Now, having spoken about the foraging ecology of vertebrates in sites that have different oiling history, having spoken about the energy flow along a salinity gradient that might be imposed by a siphon, the question is, what would be the implications for fauna and flora in salt marshes? So in terms of oiling, it seems that oiling history may impart subtle effects on the foraging ecology of vertebrates. But of course, this also then just brings in, also reveals the quagmires and the need for more baseline data. And what I mean by that is the biggest challenges is that you don't have the baseline data of how the food webs were before the disturbance happened. And if anything, what our study actually contributes to is it actually brings in, it actually brings to light the need for more baseline data and more studies that actually document some of the connections that exist before an environmental disturbance. Now, the second one, salinity changes may affect the basal resources affecting consumers. And what that might do is that it might actually affect the behavior of some of those consumers. Now, having spoken about that, if we then look at oiling history, if we then look at management strategies for land loss, all those things bring about a very big question. And that question is, if we're looking at all these factors, how do all those factors affect the fishermen and human livelihoods as a whole? Because we need to realize that the ecosystems that we're looking at also have a huge human factor, where a lot of humans rely on fish for their livelihood. So the question now that remains is about looking at those factors and how those factors may actually affect human livelihoods. And this really brings us into new research that we will be working on at LSU, and with three of my collaborators. And mainly what we will be looking at is trying to look at this human aspect of coastal restoration and how that can actually affect recreational fishing, and how that can affect the vulnerability of the people that depend on those fisheries. So pretty much what we're looking at is we have five broad questions, but I'm just going to focus on four questions, and I'm just going to make that as a broad stroke as possible. So the first thing that we want to look at, we have already been working on characterizing the ecosystem structure and function. But generally, if you look at the data that's available, it hasn't been put um, one one database where we can look at all this data, given all the data that has been collected to date. So one thing that we're going to start by doing is we're going to characterize ecosystem structure and function using isotopes, but we're actually going to feed that isotope data, which there's a lot of. We're going to feed that into ecosystem models like EcoPath. And I'll be collaborating with Dr. Polito on that. The second thing that we're going to look at is we are going to see if we can look at the current habitat suitability models that mainly use LDWF data, but they don't use citizen science data. For example, where does a fisher go? They don't have those kinds of data available. So our plan is to assess and update these fish habitat suitability models using citizen science and also using fisheries data. And pretty much what we're doing with citizen science, we're using an app where we'll actually go and talk to fishermen and actually get an idea where we've got this app called Fish Data for App that we got through CRCL, and we'll be using that app to kind of get an idea of where the fishermen are going, what they've collected, and also that will give us an idea of where we'll then use that data to then feed into the actual ecosystem model to be able to predict where the fish might be moving and where they might be going in the future. And the second thing is we're also then going to characterize and assess the vulnerabilities, the adaptive capacities and livelihood strategies under environmental change. So one of the questions that we'll be addressing there is we're going to be looking at, what are the adaptive strategies that these fishers might probably use in the future? What are they going to do if they cannot catch as much fish? So this will also then allow us to kind of assess vulnerability, but also to look at their adaptive capacity. And then at the end of it, we hope to then put this together and look at the actual socioeconomic vulnerability. So attach an actual monetary estimation of how that might affect their livelihoods in terms of a dollar value. And with that, I would like to thank everyone that has really helped me collect all this data. It wouldn't be possible without all these many people. There's so many people that have really helped and put this project together. So thanks to all of them. Thank you very much.
All right, thank you so much. It's been great. So we also have a lot of great questions. So I think I'll start with the earliest question I got. This one's from Jill. By the way, you guys can hear me, right? Just want to confirm. Can you guys hear me okay? All right, great. Thank you. So Jill asked, how did you choose which end members, i.e. the basal resources, to use for your baseline?
Okay, so we kind of chose, so generally based on other studies where other people have looked at, we knew that C4 plants were baseline. We knew that, it's a C4, where there's terrestrial plants, it's a C4 based food web. And we also knew that, and POM generally is a collection of all algal sources, right? So pretty much those two baselines pretty much give you that. We knew that there were no C3 plants, it was all C4 then. So it's all C4 plants. So generally, Spartina is a good C4 plant, is a good baseline. And then also looking at POM, which is mostly algal. So those are the two baselines that we had.
I have a sort of beginner question here. So I've never taken bio or anything, but so the C4 plants are like trees with trunks and things like that. The C3 plants are like what, reeds, corn, that kind of stuff?
I guess more, I think C4 and C3 is more of the photosynthetic pathway, but I don't know if it always holds true whether the ones that have trunks are always C4 or C3.
Okay, so it doesn't exactly match with morphology.
Huh?
It doesn't exactly match with morphology of the plants.
I don't think so. But you know what they say, don't ask a zoologist a botanist's job.
Okay. I have one more question.
I'm not really sure, but I don't think it's something to do with morphology, but something to do with the actual photosynthetic pathway that goes on within plants.
Okay. I was wondering about particulate organic matter too. Like, so you said that one is mostly connected with algae. Is that right? Yes. So how do we know that there's not like
so though we didn't mention it here, so what they did is after you get it on a filter, so you collect it on a glass fiber filter, you do look under the microscope to see to remove invertebrates and everything else, and you actually see how much algae you have there.
Oh,
So
okay.
the people that collected the data actually looked at that. So we're confident that most of that POM was mostly algae.
Okay.
Of course, you will have other things, but most of it was algae.
Sure. Okay, so Sarah Wang asked this question. It said, "Hi Sydney, would your samples allow you to detect seasonality in the diets of sparrows and rats, or the samples only collected once a year, same time every year? And is this ecosystem strongly seasonal? And would you expect to find any interesting variation in diet due to this and its interaction with oiled and unoiled sites?"
Okay. I think there wasn't a seasonal aspect in the sense that most of them were just collected in the summer. There were some samples that were collected in the fall for rats, and when you look at some of those, they don't really differ with the ones that were collected in the summer. So seasonality might have been a factor, but more the comparisons that we're making were more interannual as opposed to those seasonal differences, because those were all the samples were collected in the summer, except for those rats that were collected in the fall. But looking at them, they were not very different from rats collected in the summer.
Okay, this next question is from Dr. Linda Bowie. She says, based on the CSI, do you think the spiders in freshwater areas are eating fish since the other insects that might be their prey are not changing?
Repeat the question, sorry.
Oh, okay. So she said, based on the CSI, do you think the spiders in freshwater areas are eating fish? And the motivation for the question is that it seems that the other insects that might be their prey are not changing in the CSI.
I see. Um, I've seen spiders capture small fish, especially a lot of fishing spiders, which I've also seen on the marsh. So we do know that they can catch small fish. We do know that, but that's a possibility considering, yeah, the insects are not changing very much. That's a good point. I think because we know that they can catch fish. We know that for a fact because they do move around. So it might be that's a good point, Linda. I think that's definitely a possibility.
So oiling might be impacting their selection of fish versus other things, right? Is that the
Well,
idea? Or
well, since spiders we're really using spiders more on the salinity part, we didn't collect a lot of spiders for oiling. That might be, but we could look at those particular sites because like PS5, we could try and compare those sites. But oiling might affect that, but didn't really look into that too much.
okay, this next question is from Ian Zink, and they said, near siphon sites, there are more aquatic contributions for fish and crustaceans. Could this be related to nutrient introductions, not salinity, as you move closer to the siphon, i.e. More algal production, less food limitation, and further from the siphon, they might need to rely more on detrital food web with more contribution from terrestrial plants?
That's a good point. That's true. It might be that salinity might not be the main driver. Um, the other thing, it could be the type of vegetation, that you have, which we still need to obviously kind of characterize, and that can be a bit challenging. So yeah, it's very possible that that might be what's driving. But at the end of the day, there is a salinity gradient going on there.
Perhaps salinity is a good proxy for nutrient gradients as well.
Yeah, I think so.
Okay. And this one's from Chris D'Elia. He said most plants are C3. Oh, I think this is just a statement about my C3, C4 question. He said most plants are C3. C4 plants grow faster. An example would be Spartina. Okay.
Okay, good to know.
Thank you. Okay, so any more questions? That's the end of my question list. But happy to take more questions. It was a great talk. Okay, I think that's it for now. So just want to thank you again. Yeah,
question,
Mike.
More of a comment and a thank you. I want to thank Sydney for the speaking and for Robert and everyone at SEGO for organizing this. I do want to mention that Dr. Moyo, while he was here, he contributed and gave a lot of guest lectures and really helped us redesign our stable isotope biogeochemistry class, which is a graduate level course. And that's something that I know he has a lot of teaching responsibilities in his new position. But I'm hoping that the next time we teach that here, that he will once again grace us with his ability to give some great guest lectures, especially when it comes to a lot of teaching our students a lot of the the modeling techniques that he just the isotope modeling techniques that he used for this presentation. I know he did work a lot of our students through those isotopic mixing models and things in class, and that's something that I know that he and I have talked about, continuing that connection between him and LSU and our department moving forward.
Thank you very much.
Okay, great. Thanks everyone for turning out, and hopefully I'll see everyone here in the spring and maybe even in person.
All right. Thank you so much.
Yeah, thank you. Take care.
Thank you.
All right, bye.
Sydney.